let's do it. So, hi everybody. Delighted to see you've all come at this slightly unusual time. A slightly unusual time is to fit in our fantastic overseas speaker who's speaking from dawn in Bali, as I understand it. So, this is really quite amazing when you think about it. So, welcome to another Genomics Aotearoa seminar. We're really pleased today to have a fantastic overseas speaker, and I want to start by just thanking Dr. Ann McCartney, who's just got married in Ireland. So Ann is here. Ann used to work as part of Genomics Aotearoa and is now doing awesome things at the NIH, and so she organised the seminar. So thank you hugely, Ann. That's really helpful of you. And I'd just like to introduce briefly our speaker. So our speaker is Dr. Karen Miga. She is an assistant professor of biomolecular engineering at UC Santa Cruz. Her work focuses on genomic and epigenetic structure within human centromeres and constitutive heterochromatin. It all sounds very complicated. Effectively, she's lauded around the place for being one of the people who are really filling up those more difficult regions of the human genome to sequence. And she'll be talking about that work. She seems to be very well noticed by the international media. So was one of the 100 most influential people in 2022 by Time. I think that's Time magazine. And also, Nature claims that in 2020, you are one to watch, one of Nature's top 10 scientists around the world. So really, really fantastic to have you here, and particularly as you're presumably on holiday in Bali. So Karen, thanks for offering to present, and please take it away.
Thank you so much for the introduction, and thank you, Anne, for putting my talk on the schedule here, because it's wonderful to get feedback from this particular group. As introduced, and thank you for that, I have spent my entire career thinking deeply about human repetitive sequences. And so what in the world am I doing standing before you in this virtual platform talking about a human pangenome project? I will say that a lot of the new technologies with genome sequences and genome reference work has now put us in a really strong capacity to begin to study reference genomes in the context of human population data, and starting to look deeply about generating a human reference genome that best represents our species. And that's really the message that I'm trying to get across in my short talk today. So when I start, I think everyone on this call clearly understands the benefit of a human reference genome. We've celebrated having the human reference genome for 2 decades, and it was largely an international collaborative research program. And the key goal here was to actually represent all of the genes in a human genome and build this kind of scaffolded map. But the important part that I want you to walk away from is that it's not just a series of A, T, C's, and G's that has made this triumph so important 20 years ago. It was more of a release of a reference genome, or a set of coordinates that have been shared internationally across many, many different researchers who have started to populate and annotate and add information base by base to this map. So we've learned how variation maps in the coordinate space of this map. We've started looking at important clinical variants in the space of this map. And we've also begun to look at just basic biology and understanding how our genome works. Now, there's some really fundamentally problems with this map, in the sense that we all know that it largely represents the genomic information of a single haplotype or a single human genome. What I'm trying to show you here on the left-hand side is HG38, which is our most recent reference genome before we released the T2T genome. And the breakdown based on the ancestry of that particular individual demonstrates that the reference genome is largely 58% European, 37% African, and then 6% Asian. And by having a single haploid reference genome, we're now faced with a lot of challenges in the genomics community, such as mapping biases that are due to variants that may be in the individual's genome you're studying, but not present in HG38. There's large structural variants and inversions and complicated regions that often span repeats that may not be present in HG38 or may not be mappable within the context of a single reference genome. And then ultimately, we've been, as I mentioned in my previous slides, just really pulling in a tremendous amount of data across the world. And it's just a technical challenge to be able to study that amount of data and that amount of variation and visualize it relative to a single reference genome. We also have a problem in human genetics and genomics outside of reference genomes, where even though we've started to look at the distribution of ancestry around the world, we have a disproportionate amount of European genomes that are commonly being represented in our genomic reference materials, such as I'm showing you here for the GWAS studies. And so this is something that we can also argue that our current human reference does not properly represent global genetic diversity, nor does it really set the stage for the next phase of human genetic genomic research, where we're aiming for a more balanced and equitable platform for studying human genomes. So that's all to provide some sense of an introduction of the Human Pan Reference Genome Consortium. And in the HPRC was really a call to action from NHGRI. The idea here was that we were going to take a step further than what was promised by the original Human Genome Project and try to improve the representation of a panel of at least 350 individuals that represent both haplotype or genetic diversity and biogeographical diversity. In doing so, we have taken even a further step on a technical angle of saying, look, we want to aim for the most complete, most comprehensive maps of genome variation. And then we're tasked with trying to put these 350 genomes together in a manner that actually makes it useful for end users to study a new data structure, an ecosystem of tools in order to use this massive new pangenome. And so all of this falls into the goals and where we're trying to go as a consortium for the Human Pangenome Reference Consortium. So the question we often get asked is 350 genomes sufficient to meet this goal? When we started it, this project, this was the number that was given to us from NHGRI. It was a theoretical estimate, mainly based from single nucleotide variants from the 1,000 genome study, where we could look at the variant spectrum and of common variation in the mappable regions of HG38. And what was determined at that time was that 350 would perhaps be that special number that would allow us to capture common variation in the human population. However, we understand as a consortium that this perhaps is a flawed estimate, and the fact that we won't be able to really assess that this is going to be the true number because we're not able, as I mentioned before, to truly capture all of the variation due to the mapping biases, due to our inability to actually identify and target the structural variation, which could be large. We also know that this diversity is uneven across the genome. There are certain regions that we know are perhaps highly conserved, that may be easy to map and analyze in terms of variation, and others that may represent large tandem repeat classes or segmental duplications, which may have a more dynamic evolution and present more variation in the human population. Also, we know when using short reads, we're going to have inherent biases, such as GC richness and repeats that just won't map well, that will have their own level of variations that are technical. So overall, we think as a consortium, this is likely not true. We're likely not going to be able to capture all of human variation with only 350. So what I wanted to set your mind with at the beginning of my talk is that a lot of what we're doing in these first 5 years may be setting something akin to what was done with 1,000 genomes and establishing a technical foundation of not only the groundwork of trying to build a consortium that's capable of doing this with ethical teams, population genetics, people really lean into the technical assembly work, but also all of the pangenomic tool development and outreach that needs to go along with it. So there's two major points that I'm going to really focus on in my talk because I think that they're important to the mission statement of our consortium. And that is that we're trying to capture diversity. Diversity here means genetic or haplotype diversity as well as biogeographical diversity. And also we have an equal charge of trying to release extremely high quality, complete and phased reference assemblies. And the reason that's so important is because if you're looking at this cartoon and every single individual could be sampled or represent an individual that's a participant in our study that's been sampled from the world that represents a haplotype which offers us diversity and common variation. Once we get through our assembly efforts, if you see those two lines, one is gray and one's black, one would represent a complete gapless assembly of the maternal genome, and the black would be the complete and finished copy of the paternal genome. And that's really important because we're trying to aim comprehensively across all genomes. And it could be extremely problematic if I showed you a very different picture, where all of these lines were somewhat patchy and the orange and the red had less representation and less levels of completeness, because then you can misinterpret variants that exist in one genome versus the other. So really this emphasis on quality is really the flagship of our particular consortium. And what we're doing is taking this panel of finished reference genomes, as I'm trying to show you with these cartoon lines, and reformat them into something called a pan genome. And so this is kind of a departure from what we've been familiar with, a linear genome, in the sense that regions of the genome that perhaps are shared across multiple individuals or shared haplotypes can go into a sequence graph or a shared node that I'm showing you here, and places where individuals are different can be identified as being these edges or variants that exist between the different nodes. And so I'm trying to highlight this by showing you copy number variation, where you could have the gain of a node in only certain individuals, single nucleotide variants or SNPs, where you can see a single base being different, and even at the range of inversions. We're trying to monitor the whole spectrum of variation across these highly high quality reference genomes. And so to do that, we really have to have a multi-platform approach, where we've been trying to throw the kitchen sink at this problem at the very beginning, utilizing some of the strengths of long read platforms such as HiFi, ONT ultralong, which I'll talk about briefly. We also use chromatin capture or Hi-C with the Dovetail platform from OmniC. And we've been toying with other data types such as bionanomaps, Illumina, and StrandSeq in order to really create a whole repertoire of tools to try to build the best references possible. Now, two very important data modalities to our work is been benefiting from these long read sequencing, one being the PacBio HiFi data, where many of you may be familiar, but this is a highly accurate, almost like a Sanger quality read that can reach the lengths of 18 to 20 kilobases. And so this is an amazing tool for our team, because we were able to identify and really utilize some of these small variants that can differentiate one repeat unit from another and traverse across very difficult and complex regions of the genome. And the way that it can be so accurate is if you look at a sequence and you consider it in a circular DNA, and you had a polymerase that was going round and round, essentially you're creating a series of subreads, and each of these have errors. But when you look across the subreads, when you're sequencing the same sequence over and over again, you can take a consensus, and this consensus is highly accurate. In addition, we've been optimizing methods here at UC Santa Cruz to increase the number of what we call ultra-long reads. This is when you are feeding DNA through a protein pore, and you're able to sequence across by measuring the signal displacement and looking with some really fantastic base calling software to predict the bases as they go through this pore. And there's no real length limitation here. And so we're capable of now reaching reads that go up to megabase in length, as well as routinely reaching sequences that are 100,000 kilobases or greater. And so doing this work is big science at its best. We've been working on a multi-platform as well as a multi-center approach, where a lot of the cell lines that we're working for are all in production and lot through Coriell. They're shipped out to our various centers after QC, and then we go through massive sequencing consolidation on Amazon cloud platforms, and then we establish our reproducible workflows. And so we're really looking at a huge technological challenge that hasn't really been addressed before a consortium stepped up, which is how do you not only produce a genome that exceeds HG38, but how do you do this at production scale? And so we've released our first paper talking about our year one assemblies, and what we were able to do for that particular paper is demonstrate that although we're not yet to the finish line of complete and polished and finished assemblies, the assemblies that we have are pretty spectacular. And what I'm trying to show you are all of the assemblies on the top right panel, where you can see in red and blue are the female and male haplotypes, and where we're able to actually show that we have a lot of contiguous or comprehensive surveys of a genome reaching the same amount of sequence assembly as what we're observing in the first finished human genome, which I'll talk about briefly. The one exception, of course, your eyes are jumping to is where you see the box plot go down, but that's completely expected because that is the haplotype which contains the Y chromosome, which we expect to be of a shorter length than the X. And so that was a really lovely control. We also are seeing consistent quality across all of our reference genomes, It doesn't matter where we're getting our data from. We've now standardized all of our production methods across so many centers to where we're getting really lovely and consistent results, which marries to our bigger goal of being able to be comprehensive across our comparisons to detect new variants. So a lot of this effort is really credited to a massive team, as I tried to show on the other line. There are two major centers. One's a reference production center, which is based at UC Santa Cruz in the States, which is where I'm physically based, as well as a coordination center, which is at Washington, St. Louis. And we've established a working group structure, which I'm trying to summarize here, which really works through the identification of samples through our population sampling and representation working group, the technology and production efforts where we try to identify coverage, best practices, QC metrics, and bringing that all together. We have dedicated teams of experts that are working on just phased assemblies and trying to put the pangenomic technical components together. And then there's a tremendous amount of attention to data management, production protocols, and sharing on cloud platforms. And why I wanted to put this slide in particular was also to emphasize that throughout all of the different working groups that we have an embedded ethics and policy group that's actually are integrated, and they are able to give us rapid feedback as we move through, because this is a very interesting project in the fact that it's trying to generate a resource for the world, but it's really at the intersection of a lot of questions that could be important for having ethical and legal consideration. And so here I'm just trying to give an overview of our working group structure out of consideration that I know that Alice Popejoy will be talking to this group later, and my role in coming before her was really to give an overview of our consortium so that her talk could be more meaningful. And here I'm trying to demonstrate the more detailed view of our working group structure. And once again, the little icons on the bottom are really highlighting what Alice is going to indicate later in her talk, where we have kind of an embedded ethics model where each one of these now has kind of an ethical specialist to help us review some of the actions as we move forward. And where I'd like to go with this talk near the end is that we have been focused so much on a lot of the technical components that we are now starting as a consortium to look outward and to start to expand our international engagements and also outreach with Indigenous communities. So as kind of an overview of the remaining part of my talk, I wanted to not only go over some of the inclusion that we are doing for samples of population sampling and representation in order to improve the representation of diverse human haplotypes. I also wanted to briefly touch on our goals for future international engagement, and then to briefly go over at the end where we are now in terms of reaching this technical goal of complete and finished T2T assemblies. So for the Human Pangenome Reference Consortium, our phase one, we actually started this consortium right at the beginning of the pandemic. And so it was quite convenient that we could utilize cell lines that were already available to us at Coriell from the 1,000 Genomes. And the 1,000 Genomes Consortium, as many of you may know, really aligns with the goals of the HPRC and the fact that all of the cells have open and unrestricted access. We knew that there were already studies that predated our consortium work that studied normal variation, where the goal of that consortium was actually to represent 1%, 95% of variance with 1% frequency in the population. And this really is a resource that's been used by many people in the community to really study association, platforms for sequencing, and really establishing best practices for genome sequencing. In our most recent bioRxiv preprint, we've released 47 of these reference genomes, or 94 phased assemblies. And what I'm trying to show you here is the breakdown of those individuals that were in our first year one release. And what we were aiming to do is cover both genetic and geographic locations around the world, as well as really emphasize the technical components of this, where we are selecting cell lines that are low passage to avoid spurious mutations that could find a life by extensive cell culture. And also we were initially aiming to just include individuals that were children of trios or had parental data that we could use to confidently phase the maternal and maternal haplotype. So if we were to take a t-SNE plot and actually plot out the 1,000 genomes, we as a population sampling and representation working group could begin to assess how we are performing in terms of having coverage of the 1,000 genomes and representation of the variants that we know are inherent in this particular data set. We also paid a lot of attention to known haplogroups, such as the Y haplogroup, where we tried to increase and ensure that we had coverage of different Y haplogroups from the 1,000 genomes in order to be able to do some really lovely studies, specifically focused on sex chromosomes. Now, hopefully many of you on this meeting are quick to realize that the 1,000 genomes alone is insufficient to fully represent genomic diversity within the human population. We would all agree with you. And so this is a moment where we're trying to expand our consortium to not only go into prospective recruitment, but also to start to establish global genomic partnerships. And this is largely being performed through an alliance with the Global Alliance for Genomics and Health, or GA4GH. So when we think about improving population sampling and representation, as I mentioned before, we've been solely focused on the cell lines that are available to us for the 1,000 genomes. Our target here is to reach 200 or 350 of these particular available cell lines. We are aiming to move away from our dependency on using trios or the parental data. This was something that was shaping our ability to fully sample diversity within the 1,000 genomes. We are now in mode of launching new recruitment efforts in the States, which I'll talk about briefly, which will allow us to study and bring in participants from different areas that perhaps could present new variation that was missing from the 1,000 genomes efforts, and also through making new collaborations and international partnerships. So our next phase 2 is really to look at population representation and sampling. And in this case, what we're doing is we're working to set up a new recruitment center with BioMe Biobank. This is out of Mount Sinai in New York, where we have greater than 50,000 participants that now exist within this domestic biobank, which present a wide range of genetic diversity and also self-reported ancestry. We also are teaming up with the Washington St. Louis Recruitment Center, where they have been working now for over 2 years with a community of African Americans in the area who are working towards common disease reference materials. And so this is another opportunity for our consortium to partner and try to share the methods and the goals of what we're trying to do and recruit participants to our study. I wanted to quickly go into the BioMe Biobank. I thought this might be of interest to folks on the call. Essentially, the BioMe Biobank, as I mentioned before, is based in New York. This is one of the most, diverse areas in the United States, where we expect to have a large percentage of non-European ancestry participants, as you can see in this dot plot, where the size of enrollment is shown by the circle, and then you have the percentage of non-European ancestry on the y-axis and the year of enrollment on the x-axis. Overall, we have over 60,000 of these participants that are currently enrolled. I have the breakdown of their self-reported ancestry, as you can see in the pie chart. We benefit from having genomic data sets on exon sequencing as well as whole genome sequencing and array data. And we have a consent that's specific to allow us to recontact for future research. So now we're going through all of the careful protocols to ensure that we can share information about our consortium and start to invite new participants to join through the BioMe project. And as I mentioned before, New York City is rich in complex diversity. So this is really trying to illustrate that we're not looking at the same type of clustering as we had with the population labels that were used before for 1,000 genomes, but rather a continuum representing a lot of admixture that exists in New York, but a lot of diversity yet that still may exist outside of the 1,000 genomes. Of course, we want to move away from only including participants from domestic biobanks in the United States. So this has also launched a very exciting phase of our consortium where we're hoping to bridge with international partnerships. In doing so, we've launched something called the Human Pangenome Project. And it's really an opportunity to form an international alliance with global genomic partners with the common goal of providing accessible high quality genomes to represent diverse human populations. So this, once again, is taking a lot of the early work that came out of our consortium and trying to move it to an international stage to where our team would be one of many sitting at a global table working toward a resource which we hope will be extremely valuable for the global genomics and clinical communities. And so doing so, we're hoping that this reference resource will be inclusive with the goal of a common pooled resource for humanity. And doing so, we need to develop strategies to evaluate and measure gaps and also explore any opportunities to ensure global support. We're also hoping to improve clinical genomics and biomedical research globally. So a lot of the tooling and new infrastructure that we're building needs to be attentive to that type of bridging with other consortium efforts to ensure that clinical data sets and clinicians have access and outreach and training to utilize this resource as well. And so this equitable training and outreach is something we are also taking quite seriously to ensure that and then sorry, I don't know why it's doing that. Let's see if I can close that. Sorry, so this is really aiming to have genomics outside of our United States efforts. And so we're looking at the Human Pangenome Reference as being something that we're going to align once more with GA4GH. And then uniting with the beginning of my talk, we're hoping that this will be that new coordinate-based system that will be shared internationally, will be the new foundation for understanding human variation in the future, clinical, and how our genomes work. And in doing so, we're actively forming these new partnerships around the world and trying to come together as a community to begin to work on building this global resource together. So with that, I wanted to also kind of give an update to this team on where we are in terms of assembly T2T technology. What does that mean and where are we now? So when I first introduced the Human Pangenome, I really wanted to emphasize two major components. One is the image that I'm showing on my left-hand side, which is, collecting and representing diversity around the world. And on the right-hand side is this technical goal, which is to get the most complete reference genomes possible to ensure a complete comprehensive variant map of this pangenome structure. And in doing so, this introduces a real technological challenge because we now have to go through at scale to try to reach hundreds of genomes which are at this quality. However, there's another problem that I didn't introduce at the beginning of the talk, but rather that we are aiming to analyze and generate complete human genomes. And when we first started this consortium-based effort, we all knew that the human reference genome, or this HG38, was roughly 8% incomplete. And a lot of these huge gaps that we knew existed in HG38 were spanning regions that could be very interesting and important to our consortium to include. These are duplicated gene families, centromeric regions, which we know are important for chromosome segregation, entire acrocentric short arms, and subtelomeric regions. And so it's really important to include these regions to not only understand how our genomes function in terms of centromere, knowing how the genomes are faithfully duplicated and replicated, and we have a faithful division of our chromosomes every time the cell divides. Also our ribosomes that we're missing on every acrocentric, which we know are fundamental to life for understanding protein translation. A lot of these regions that we were studying that have been missing from the map are known to be very specialized in the way that they're organized. And this specialized epigenetic regulation seems to break down as we age or have cancers and diseases. So by not including these sequences, we kind of we lose the ability to actually study genetic and epigenetic variations that could be important for human disease. And of course, these large unresolved segmental duplications that we knew existed in the arms of human chromosomes could present information of studying genome instability and gene family expansions. So this was really a call to action for another consortium, a sister consortium of the HPRC, if you will, called the Telomere to Telomere Consortium, which was co-founded back in 2019 by Adam Philippi and myself. And here I'm just using this slide to really introduce it. This was a grassroots effort. There was no call for funding for this particular challenge, but yet 100 people from around the world came together during a global pandemic with a singular goal of how can we put together a finished human reference genome. And so we're being celebrated now because we were able to reach this particular goal. I'm not going to have time to go into all of the different results that we were able to present, and not only releasing a complete human reference genome, but also the 5 different studies which were able to really bring out new biological insights by having a complete genome for the first time. And so when we started the HPRC, or the Human Pangenome Reference Consortium, the T2T was developing highly innovative methods to meet that goal of how can we generate a complete, finished, and accurate assembly. And so in doing so with our first genome, we were able to show that we were able to introduce roughly 200 megabases of new sequence, which was missing from HG38. We released a whole panel of new gene predictions, 99 of which were protein coding. And we had the first complete maps of centromere segmental duplications in the acrocentric short arms. But really, relative to the HPRC, what we had established was an amazing team of long-read innovators, where scientists could come together and start to think about innovative new models for assembly, chasing perfection. How do we start to do an assessment of quality of reference assemblies? How do we do automated and annotating gene within these highly finished reference genomes? How do we actually improve the sequencing technology to work toward better reference genomes? And how do we begin to do improved variant calling and epigenetic studies within the context of a finished genome? In crediting a lot of our assembly experts who have been working on this long read platform, what we found is both the HiFi and ONT data, which I introduced at the beginning of my talk, were really critical to make this goal. And the fact that the HiFi, or the very high fidelity reads, could be used to have overlaps, to identify these perfect overlaps or create its own string graph. And what I want your eyes to see in this particular image is that all of the individual human chromosomes form their own connected component at this point. And there are regions where you can see that there are complexities in terms of interchromosomal or places where chromosomes are coming together, like the acrocentrics, or giant bubbles where you have regions where you have a duplication on chromosome 9 that were really useful for our nanopore, these very, very long reads to help work through and have our assembly team try to disentangle. And throughout it all, we had to advance tools in order to chase perfection. So it was not only enough to develop new assembly methods, but also to develop a whole pipeline to evaluate the quality, where we were able to release a genome that was looking at a QV value of above 70, or expecting 1 error every 10 million bases. So now that we have a human genome that's finally complete, what does that tell us in terms of new insights for human health? We're finding that this will help us improve clinical genomics variant calling. It improves our ability to study gene families more comprehensively, repetitive regions such as centromeres to study their function, and entire short arms. And so this target for the T2T consortium, now the shared target with the Human Pangenome Reference Consortium, is to build this more accurate reference across 350 genomes. And the reason why this is so important is because when you start looking at these highly accurate references, we overall see an improvement in short read mapping. Here I'm trying to show you that we have an improvement of mismatch rate, where the new assembly, the one that's complete, is in orange and the older one is in blue. And you can see there's a shift toward the bottom, representing a lower mismatch rate across the board. We also find there's lower quality, less Mendelian violations, and these nicer T2T quality genomes. And overall, we're finding less false variants. We now have high resolution studies of entire acrocentric short arms, representing over 60 million bases of new sequences, including new genes that hadn't been studied before, to really bring into the context of discovering new structural variation and variation that could involve human clinical disorders such as Robert's immune translocations. We have the first high resolution maps of all human peri and centromeric regions, and are able to study the satellite DNAs and the embedded transposable elements and genes within this region at the base level for the first time. Additionally, we have now complete maps of duplicated gene families or segmental duplications. This is over 81 megabases of new segmental duplications were brought out through this initial study. And in red, what you're seeing here are all the new genes that are present in stage 13 that were missing or absent from the previous genome reference assembly, and in black are regions which were shared. However, the orientation or structure of that particular gene was in a different order and organization than what we were observing in stage 13. And once we have these more complete maps, we could then take these long read elements and begin to look at the fifth base. And what I mean by that is when you can read the DNA through a pore, for example, a nanopore, you begin to detect the methylated bases, in this case CpG, and you can do it with exquisite sensitivity over very large stretches of our genome, sometimes spanning over 100 kilobases. And so by doing this, we now have a single cell, or each single molecule represents an instance in a cell, so we can also observe epigenetic heterogeneity. So in doing this with our more complete genomes and our very long reads, we're not only able to capture more complete variation maps with short reads, more comprehensive maps of repetitive genes and satellites, but also build on top of this map a whole rich layer of epigenomic data. And so, as you can imagine, just to get your eyes to focus on the methylated versus unmethylated CpG patterns that I'm showing you, In the transposable element, this is a repeat element that we expect to be highly methylated. You can see that it's largely red or represents methylated CpGs. However, in a gene body where we expect it also to be methylated, at the site of transcription, we can begin to detect this really striking strip of blue, which now gives us information about unmethylated CpGs. And so we can begin to move throughout the genome now, end to end or telomere to telomere, and begin to make these expansive maps, not only of utilizing the Hi-C data, as I'm showing you here, to illustrate that we have a massive domain structure that had been previously published and described on chromosome X, but also looking very closely at these sites where we expect something interesting to happen at the DXE4 locus, where we're able to now look at these highly detailed epigenetic patterns with the oscillation of red versus blue. So to end my talk, I think it's important to really credit where we're going, which I spent a tremendous amount of time at the beginning of my talk about setting up why we want to go there, all the goals of moving this into an international platform, and the technical challenges that are before us. And at the end of my talk, I really emphasized some of the innovation and tools and new discoveries and why it's important to have this goal of a more complete genome. However, we all acknowledge that we are not yet to the finish line. The T2T consortium is just releasing a single haplotype of an individual who's largely representative of European ancestry. And we have two technological barriers that we're trying to currently break down. One is how do we begin to transition a lot of our tools and assembly work to reach a diploid genome? And then how do we break that barrier again to the production of hundreds of T2T diploid genomes? And in doing so, we're hoping that having more complete genomes and having a better representation of genomes in both directions, both the completeness of a reference as well as completeness and representation of haplotypes, will improve healthcare by providing a more complete view of important clinical variants. Also, we're hoping that it'll provide a better lens on understanding genomic diversity of our species and open new doors to understanding cellular function as we start to look into these regions of our genome and their epigenetic regulation, where we haven't been able to really study these so close and precisely before. So with that, I'll end my talk with a pretty massive acknowledgement slide. This is one person standing in front of you, but representing a lot of work across many centers, as well as really crediting the Telomere to Telomere Consortium, as I mentioned before, which is a grassroots effort to complete the first human genome, that is now part of our pan-genome community to try to make this a model for how we can improve reference genomes for more diverse ancestry and haplotypes.
I'd just like to thank our speaker again, Karen. That was a really, really awesome talk. And I think I agree with you that this is definitely the direction we need to be heading in. And it's nice to see that actually many of the things you're talking about are tools that actually we can access, data we can generate. It's not completely out of reach. And that's
not at all.
That's really heartening. So thank you so much for your talk. I'll do again the clapping. And thank you for coming today to listen to Karen. Thanks again to Ann for her organizing the seminar, and we will see you again in a couple of weeks. So thank you very much. And
thank
goodbye.
you very much. Thank you.